Hi, welcome to Microbiology Mentor. Today we will discuss an important topic, culture media. The learning objectives which will be met today after the completion of this session will be to define culture media, explain the importance of culture media, how to classify a culture media and the types of culture media with example. So let's see how do we define a culture media. A culture media which is usually required for the cultivation of microorganism contains a combination of ingredients that will support the growth of cultivation of microorganism by providing all the essential nutrients required for the growth. So let's see what are the need for a culture media. So culture media may be required in cases to isolate the microorganism when it is required to isolate or to culture the microorganism, or it may be required to separate pure culture from the mixed flora. Also be required for identification of a microorganism, like in case of the various biochemical media which are used to identify a microorganism. Similarly, culture media may also be required to determine the antimicrobial susceptibility pattern, like the commonly utilized laboratory culture media MHA is used for the antimicrobial susceptibility testing. So for any culture media, there should be certain basic requirements, which includes the nutrient which serve as a source of energy, carbon and nitrogen. Apart from it, there are certain mineral salts like sulfate, phosphate, chlorides and carbonates of potassium, magnesium and calcium. Apart from that, there should be a need of a suitable culture pH of the media, which is usually set around 7.2 to 7.4, as well as certain accessories growth factor as and if required for a microorganism, like in case of Haemophilus influenzae, where there is an additional requirement of factor X and factor V. Now for any culture media, the constituent includes water, electrolyte, peptones, which are the partially digested proteins, then agar, which is used either to solidify or make a media semi-solid. Depending on the concentration of agar, the media can either be semi-solid or solid. For example, 0.5% agar may be used for a semi-solid consistency of culture media, whereas 1% agar is generally required for solid culture media. Then meat extract. So let's see how a culture media can be classified. So a culture media can be classified in several ways. Like, based on the consistency, it can be either classified as a liquid culture media, semi-solid media or a solid media. So blood agar is an example of solid media. Then sim media, which is used as a composite media for identification, is a semi-solid agar, or any motility demonstration media can be a semi-solid media, whereas BHI broth can be considered, or any broth can be considered as a liquid culture media. Then culture media can yet be classified on the basis of requirement of oxygen, like it can be classified as aerobic culture media or anaerobic culture media. The classical example of anaerobic culture media is Robertson cooked meat media, whereas aerobic can again be any media like blood agar, chocolate agar, McConkey or a nutrient agar, which can be which serves for the growth of aerobic microorganism. Then culture media, on the basis of the use, it can be classified either as a routine culture media, or it can be defined as a synthetic or defined culture media. So let's see first what can be the routine culture media. Routine culture media can either be a basal culture media, an enriched media, enrichment media, or it can be a selective media, or it can be a differential media, transport media, or an anaerobic media. Here it is worth discussing that a single media can fall in more than one category of the media. So let's see the example of each culture media which is used routinely one by one. Like here, you can see the first media is a basal culture media. So the basal culture media, the classical example is a nutrient agar, which is a solid media, and the liquid basal media can be peptone water or nutrient broth. So what is the feature of a basal culture media? So it provides the ingredients for the growth of non fastidious microorganisms. So it is the basal media which is providing the nutrients or enough nutrients for a non fastidious microorganism. Then enriched media. So enriched media is made by adding certain components to the basal media. So the basal media act as a base for enriched media, to which certain components like blood, like serum, or egg, which can be added to a basal culture media to make it enriched media. For example, in this slide, you can see it is showing an example of enriched media like blood agar, where to the basal culture media nutrient agar 5 to 10% of sheep blood is added to make it enriched media. Then another example is of enriched media can be a chocolate agar, which is basically a hemolyzed blood where it is added and it can be used as a enriched media. Again, enriched media itself can be liquid media. Like the classical example is the brain heart infusion, which is the media inside the blood culture bottle. Here in the slide it is shown two blood culture bottle, one classical, one conventional, other is an automated blood culture bottle, both having enriched medium like BHI. So BHI here is an example of liquid enriched media. So let's see what is an enrichment media. So an enrichment media is a type of media which allows the desired microorganism to grow from a mixed flora. So it is usually used in cases of a sample which contains mixed flora. For example, from the stool which contains the pathogenic organism as well as the non-pathogenic commensal microorganism like E. coli. So the desired organism can be enriched using either tetrathionate broth or selenite F broth. Now let's see what can be the example of a selective culture media. So selective culture media inhibit the growth of microorganism. It actually inhibits, it has certain inhibitory components where it inhibits the growth of certain microorganism over other, like in case of McConkey agar, which act as a selective media where it inhibits the growth of gram negative bacteria as well as it facilitates the growth of gram negative. So it inhibits the growth of gram positive whereas it facilitates the gram negative bacteria. So here it is adding, or it is adding as a use of, or it is serving as a use of selective media where it selects gram negative over gram positive. Similarly, TCBS is used as a selective media for Vibrio cholerae. Similarly, LJ media, which inhibits the normal commensal of the oral flora over mycobacteria, is also acting as a selective media. Similarly, Wilson and Blair media is also acting as a selective media for Salmonella typhi. Similar example is potassium tellurite media where diphtheria bacilli is selectively grown over the other normal commensal microorganism. So here in this slide you can easily make out that TCBS on the left side LJ media in the center, and whereas Wilson and Blair media, all the three media are actually selective culture media. Now let's see what are the differential culture media. So the differential culture media, as the name indicates, it differentiate one type of microorganism from another. So the classical example is a clad agar or a McConkey agar, where both these culture media can differentiate lactose fermenting colony from non-lactose fermenting colony. So on the clad agar, the lactose fermenting colony appears yellow in color, whereas non-lactose fermenting colony appears pale in color. Similarly, lactose fermenting colony on a McConkey agar appears pink in color, while non-lactose fermenting colony appears pale in color. So now another example can be a transport media. So this kind of media retains have the capacity to or have the power to retain the to keep the viability of microorganism intact, whereas it does not allow the microorganism to grow further or multiply further. So a good transport media should allow the microorganism to remain viable, whereas it should not allow the organism of interest to multiply. The classical example of transport various transport media includes the Stewart media, which is used commonly for aerobic as well as facultative anaerobic bacteria, whereas Amy's media, which are again can be used for most aerobic and facultative anaerobic bacteria, whereas Amy's media impregnated with charcoal can be used for the transportation of glucose. Carry-Blair media can be used for the transportation of the pathogenic stool bacteria, commonly Vibrio cholerae, and buffered glycerol is again used for the transportation of potentially pathogenic stool microorganism. Now, regarding the anaerobic culture media, as I have discussed, that anaerobic culture media are those media that allows the growth of anaerobic microorganism. Here, the example on the left is shown as RCM, which stands for Robertson cooked meat media. This media has a beef heart which is rich in glutathione. The glutathione causes a lower in redox potential, making at a kind of anaerobic atmosphere, facilitating the growth of anaerobic microorganism. Similarly, on the right is another example of anaerobic media, which is thioglycolate broth, where it is supplemented with hemin as well as vitamin K. So now coming to the last type of culture media, which is said as defined or synthetic media. Here the entire exact composition or the pure ingredient is added carefully with a measured quantity or concentration, so that the exact chemical composition of the media is known. So these are synthetic or laboratory made media. So they contain a simple sugar as carbon, an energy source, and an inorganic nitrogenous source, various mineral salts, and if necessary growth factors like purified amino acid, vitamins, purines, and pyrimidines to facilitate the growth of microorganism. A classical defined or synthetic media can be RPMI 1640, which can be used for several purposes. With this, I finish the culture media. Thank you.